Yeah, so today I'm going to talk, I think Samantha touched on it a little bit about looking at the possibility of non-canonical DNA bases and the evolution of DNA bases. So the harsh UV radiation that I mentioned is possible because right now on Earth we are protected from harsher UV radiation by the ozone layer, reflecting and absorbing some of it. And so in prebiotic Earth, before the ozone formation, and also in extraterrestrial situations, we would have much more of this harmful UVB, UVC wavelengths, which could shape this evolution. So this is important because all of the canonical DNA bases absorb in this UVB, UVC region where we're essentially protected by the ozone layer, but under solar irradiance would not be. So we decided to focus in this study on the purine family of nucleobases, specifically adenine and guanine, and these two bases are shown to be very photostable. So if you shine UV light on them, they have mechanisms to get rid of that energy within hundreds of femtoseconds, and they go back down to the ground state very efficiently. So they don't populate long-lived triplet excited states, and they go back to the ground state. But the question that arises is that these have different structures, but they're fairly similar. Would this be the case for all other types of purine derivatives? So other works that have been done in the past are derivative of guanine, hypoxanthine, which it also has been observed to ultra-fast intersystem cross back to the ground state. However, just as we saw in the last talk, this derivative of adenine, and I guess of guanine as well, is actually fluorescent, and also it is able to populate a long-lived reactive triplet state depending on its environmental conditions. So this shows that it's not definitely all the purine bases that ultra-fast intersystem cross get rid of that energy on an ultra-fast timescale. So then the question that arises is it due to this purine core, or is it the specific substituents on the purine core that are moderating this excited state activity? So to begin looking at the purine itself, we looked at the steady state, just the absorption of the base itself. And we see that just like the natural bases, adenine and guanine, it has a absorption in the UVB around 260, and then a higher energy, stronger absorption, and then a tail going out to about 310 nanometers. Then we looked at its fluorescence, and we see that, like the natural canonical bases, it has very, very low fluorescence, quantum yields of 10 to the negative third, so essentially zero fluorescence. So this directly matches with the purine and derivatives adenine and guanine, but we want to know, does it ultra-fast intersystem cross like those two bases? So to do that, we use our pump probe transient absorption spectroscopy, where we use one laser pulse to pump our sample and send it to an excited state. And then we use a second pulse at a certain time delay to look at changes in that excited state. And we detect changes in the absorption of the sample, and we see the change in absorbance. So this is the typical type of data that we get as a contour plot, where we're seeing wavelength on the X here, time on the Y, and then absorption intensity as the color, with red being the highest. So we can see as when we pump the sample, it instantaneously goes to the excited state. So at time 0, we're seeing these signals of excited state absorption already. And from this type of contour plot, we can extract kinetic information and then also spectral information by looking at either slices in the wavelength regime or slices in time. So these are examples taken from that contour plot of the type of kinetic information we get in traces, or spectral information that we can see. So if we first look at the kinetic information, and we compare it to the natural adenine base, the purine core does not go back down to the ground state immediately. And in fact, in this time scale, we looked out to 3 nanoseconds with our setup, and we can see that we're actually beginning to populate even longer lived excited states at the end here that will go on for longer and longer time. So these purine excited states live for at least 4 orders of magnitude longer than the natural adenine and guanine bases. So that could be very potentially harmful for the purine core. So to get an idea of kind of where this excited population is going, we can look at the spectral information that we get from the transient, but you can see that there's quite a bit going on here, and it's fairly hard from just experimental point of view to extract what's happening. So to help us out, we turned to some of our collaborators in Madrid and in Vienna, where they do high-level computational chemistry. And so using their computational techniques, they can actually model the potential energy surface of each of these excited states. And then using also surface hopping dynamic simulations, they can predict where the excited state population will likely travel along these surfaces. So this is a pretty complex graphic, but if in the ground state we excite with a UV photon up to the S2 excited state, then the computations and the dynamic modeling predict that the most likely relaxation pathway is followed by these yellow arrows. So a fast decay to this conical intersection with the S1 state, then a rather slow decay across this somewhat flat surface to a crossing point with a triplet, and then efficient population of the T1 minimum, where it gets stuck in this triplet excited state and it really has no way to get back to the ground state. So it takes a very, very long time. So this is what the computations predicted. So now we want to try and match that up with our experimental results to see if we can put together a mechanism that's fairly concrete for this compound. So to do that, we actually chose points along this predicted surface to do vertical excitations from, like we would do with our transient setup. So when we probe, we're actually probing here and looking at absorptions from these higher excited states. So we chose these points and tried to simulate our transient spectra from these points. And so we can actually see that the experimental spectra taken at certain time delays and the simulated transient spectra from the computations actually match up very, very well and have the same type of transitions going on, which I can discuss more in detail if anybody's interested. But essentially it gave us this mechanism where that follows just as the computations predicted, where we eventually lead to a long-lived triplet excited state, which takes a long time to relax back down to the ground state. So from this, we learned that that the purine core doesn't ultra-fast internal conversion to the ground state. Rather, it intersystem crosses to populate the triplet, and that triplet lives a long time, which gives it a long time to do chemical reactions. So the core of these compounds is not responsible for the photostability seen in DNA. Rather, it seems to be that having a substituent at the 6th position is what is really key to their photostability. So whether it's an amine group or an oxo group, that provides it with that photostability. And so this lends support to these bases being on prebiotic Earth, being photostable, and also provides targets for searching for other non-canonical bases, but requires that they should be substituted at the C6 position in order to be photostable and survive harsh UV environments. And so with that, I'd like to take any questions, and thank you all.
So, with the energy levels and the surfaces, do you think that with the substituted purine, do you think there's an additional conical intersection, or is it just that the?
Yeah, we're working on that right now. Some older computations suggest that this S2 drop here actually keeps going when you have that C6 substituent. So the I guess speed of the wave packet moving across this potential energy surface kind of bypasses this crossing point and just goes back to the ground state. But we're working on that right now.
Okay, cool.
Other questions?
Have there been experiments just taking that purine core and hitting it with light and seeing how stable it is?
I'm not sure. I know that it has been, the triplet population has been measured to be in the microseconds. So I would assume it can highly react. It's triplet, so it can react with oxygen and other things in the environment. So yeah, that's another possibility. Maybe the purine core, being highly reactive, is reacting to form these photostable DNA bases. Maybe that's another route to it, but I'm not sure as exact photochemistry that's been done with it.
Could be interesting.
Any other questions? All right, now let's thank Marvin again.